Okay, so like, have you ever heard about like how fast a chameleon's tongue can shoot out and catch something?
It's crazy. Faster than a hydrojet accelerates.
Right. It's mind-blowing. And like how some of those male anglerfish, they like fuse right onto the females.
It really makes you wonder, what are the fundamental things, the building blocks, that make all this variety possible?
Yeah. And that's what we're getting into today. And we have this whole chapter, right, that kind of lays the foundation for the whole animal kingdom, like the basic characteristics, how it all evolved over, millions and millions of years.
It's the core of understanding animals, the whole enchilada. What makes an animal an animal?
Absolutely. So what we're looking at today is this chapter, and it gives us this overview of animal diversity, like the essential guide. And we don't want to get lost in all the scientific jargon, but really pull out those key concepts, the things that make you go, oh, I get it now.
Yeah.
And, give a clear understanding of like the animal kingdom from the very beginning.
Exactly. A nice comprehensive summary touching on everything from their cells and how they develop to their grand evolutionary journey. We want you to walk away with a solid foundation and a real appreciation for how amazing animals are.
Perfect. So the chapter starts by asking, what makes an animal an animal? And it all begins with how they fuel up, right?
Nutritional mode, exactly.
So
It's basic but important. Animals are heterotrophs. They got to eat other stuff, other living things to get energy and nutrients just like us.
right. Not like plants soaking up the sun all day.
No photosynthesis for us. Unlike plants, which are autotrophs and make their own food, or fungi, which sort of absorb nutrients around them, animals we ingest, we eat, and then break that food down inside. Big difference.
Makes sense. So on to the next thing, their cell structure. And it's amazing how something so tiny can be so important in understanding, like, what makes an animal an animal?
Yeah, and it all comes down to specialization. You see, animal cells, they don't have those rigid cell walls that give plants and fungi their structure. Instead, they depend on structural proteins outside the cell membrane, mostly this protein called collagen.
Collagen. It's everywhere.
Right. And it's unique to animals. You won't find it in plants or fungi. It's the glue that holds everything together, provides support.
And this lack of cell walls allows for something pretty amazing.
Tissues, groups of similar cells, all working together to do specific jobs. Muscle tissue, for movement, obviously. Imagine a worm wiggling or a cheetah sprinting. It's all thanks to muscles. And then there's nervous tissue, transmitting those nerve impulses, allowing animals to sense their environment and react.
Like when you see a yummy snack and your brain tells you, grab it.
Exactly. Muscle and nervous tissue, they're essential for the active lifestyle of most animals. They can move, sense their surroundings, hunt for food, all thanks to this level of cellular organization.
Makes sense. Speaking of snacks, you need those muscles and nerves to catch a chameleon's lunch, for sure. But let's move on to another key characteristic, reproduction and development. This is where things get more complex, right? Like, how does new life begin?
So for most animals, it's all about sexual reproduction. You've got the diploid stage, where cells have two sets of chromosomes, and that's the dominant form throughout their life. The process usually involves a tiny sperm cell fertilizing a much larger egg cell, and boom, you get a diploid zygote, the very first cell of a new organism.
And then the real journey begins. The chapter mentions this thing called cleavage. Not like splitting a diamond, though.
No gem cutting here. Cleavage in this context is a rapid-fire series of cell divisions in the zygote, but without much growth. Picture it like hitting copy-paste a bunch of times, creating tons of smaller cells from that single fertilized egg. It sets the stage for everything that follows.
Wow. So from one cell to many, and it leads to something called a blastula, right?
Yes. It's often a hollow ball of cells with a fluid-filled space inside called the blastocoel. Think of it as like a tiny microscopic balloon.
Fascinating. So we go from one cell to this tiny balloon-like structure. What's next in this incredible process?
Well, then comes gastrulation. One end of the embryo folds inward, like pushing your finger into that balloon, expanding and eventually filling the blastocoel. This folding creates layers of embryonic tissues, called germ layers, the ectoderm on the outside and the endoderm on the inside. These layers, along with a third one that forms in most animals called the mesoderm, are the foundation for all the different tissues and organs in the animal kingdom.
It's like the very first draft of the animal's body plan. And the chapter mentions that a lot of animals have a larval stage, which I've always found fascinating. It's like a whole different life before they become adults.
Right. It is pretty wild. A larva is like a teenager, not yet sexually mature, and often they look and act totally different from the adults. Like, think about a mosquito larva chilling in the water compared to the buzzing adult, or a dragonfly nymph versus the flying dragonfly.
Totally different lifestyles.
Exactly. These larvae eventually go through metamorphosis, a huge transformation that turns them into juveniles, which look more like adults but still aren't mature enough to reproduce.
So it's like a biological makeover. And speaking of makeovers, genes play a huge role in all of this, right?
They do. The chapter talks about developmental genes and how they're key to understanding the huge diversity we see in animals. You know, despite all the differences, the genes that control their development are surprisingly similar across many groups.
Really?
Yeah, there are these regulatory genes that basically act like conductors, controlling the activity of other genes during development. And a really important group is the homeobox genes, especially the Hox genes. They have specific DNA sequences called homeoboxes, and they're crucial in shaping the overall body plan, controlling things like segment identity and body organization. Imagine them like master architects laying out the blueprints.
It's amazing how much control these genes have over the final outcome. Okay, so we've got a good grasp on what makes an animal. Now let's go way, way back in time and explore their history. This chapter really emphasizes just how ancient animals are.
It's mind-boggling. We're talking over 500 million years. The chapter points out a bunch of key moments, like milestones in animal evolution. First, we've got biochemical evidence going back a whopping 710 million years.
Whoa, 710 million years. That's like almost impossible to imagine.
It is, right. So what they found were ancient sediments with these steroid molecules, the kind primarily produced by sponges, which, as we know, are animals. This tells us animals were around way before we see their fossils.
So animals are even older than their oldest fossils.
Exactly. Then we moved to the Ediacaran biota around 560 million years ago. This was a pretty strange collection of life forms.
The egg ran like from another planet or something.
It does sound a little alien. They were these early macroscopic fossils, meaning you could see them with the naked eye. Mostly soft-bodied, multicellular eukaryotes, which are organisms with cells that have a nucleus. First found in Australia, but now we've got similar fossils from all over. Some of them, they kind of resemble things like mollusks, sponges, or cnidarians, but many others are total mysteries. They don't really fit into any categories we know.
Like puzzles from the dawn of time.
Exactly. And there's this exciting possibility of even older microscopic fossils, maybe of early animal embryos from this period, though whether they're actually animals is still being debated. But it seems like a time of lots of experimentation with multicellular life.
Okay, so from mystery fossils to the Cambrian explosion, that name sounds like things got really exciting.
Exciting and fast. It happened from about 535 to 525 million years ago, a super quick burst in geological time. Before the Cambrian, fossils only showed a few animal phyla, but then, boom, we see fossils representing about half of all the animal phyla we have today, including the first arthropods, chordates, and echinoderms.
Like life suddenly went into overdrive.
Right. And lots of these Cambrian animals had hard skeletons, like shells and spines. Some like totally different from anything we see today. It was a huge turning point, marked by the rise of the bilaterians animals with bilateral symmetry, the head-to-tail body plan, and a complete digestive tract. A whole new world of possibilities opened up.
It must have been an amazing time to be alive, well, assuming you weren't on the menu. The chapter has these artists' reconstructions of the Cambrian seascape, and it's wild. You've got Pikaia, maybe an early chordate relative, the segmented morella, and then there's Anomalocaris, this fearsome predator. And hallucigenia, with all its spines, it was a whole different world. So what caused this sudden burst of evolution?
There are a few ideas out there, and it was probably a mix of things. One big one is predator-prey relationships. As predators evolved new ways to catch their meals, like claws and teeth, prey animals had to evolve defenses, like shells and armor. This back and forth probably drove a huge amount of diversification. Then there was a big increase in atmospheric oxygen around this time, which allowed for bigger bodies and more active lifestyles. And finally, genetic changes, especially in those Hox genes we talked about, might have given the raw material for all these new body plans.
So a combination of factors created the perfect conditions for the Cambrian explosion. What happened next? Did this rapid evolution just keep going?
Well, animal diversity did keep increasing, but it wasn't a smooth ride. The rest of the Paleozoic era saw some pretty significant extinction events that really shook things up. It was a time of both diversification and loss. During this era, vertebrates, like the early fishes, became the top predators in the oceans. Then, around 450 million years ago, arthropods became some of the first animals to colonize land, opening up a whole new frontier.
That's a huge leap from water to land.
Definitely. And those early land-dwelling arthropods faced a totally new set of challenges and opportunities. Then, around 365 million years ago, vertebrates followed suit, venturing onto land themselves. These first land vertebrates diversified into amphibians, which still relied on water, and amniotes, which developed adaptations like the amniotic egg that let them fully reproduce on land. We even have fossils of millipedes, centipedes, spiders, and things called fern galls, which show that insects and plants were already interacting in complex ways by at least 302 million years ago. Life on land was getting pretty sophisticated.
And then came the Mesozoic era, the age of the dinosaurs.
Right. The Mesozoic was a time of incredible expansion. All those animal phyla that evolved in the Paleozoic, they spread out into all sorts of habitats. Coral reefs started forming in the oceans, creating these amazing underwater cities. Some reptiles even went back to the water, like the plesiosaurs. On land, you had pterosaurs and birds taking to the skies. Dinosaurs, of course, ruled the land, and the first mammals appeared, though they were pretty small and probably scurried around in the shadows.
It sounds like the Mesozoic was a busy time. And flowering plants also really took off, right?
Oh, yeah. The late Mesozoic saw a huge diversification of flowering plants, and with them came all sorts of insect pollinators. It was a time of coevolution and ecological richness.
Fascinating. And finally, we arrive at the Cenozoic era, the one we're living in now. This era started with a pretty dramatic event, right?
You're talking about the mass extinction at the end of the Mesozoic, the one that wiped out the non-avian dinosaurs. It was a turning point that ushered in the Cenozoic and reshaped life on Earth. With those dinosaurs gone, mammals who had been kind of small and in the background suddenly had a chance to thrive. They diversified like crazy and evolved into all sorts of shapes and sizes.
That's a classic example of how extinctions can lead to new opportunities.
Definitely. The Cenozoic also saw some big changes in the climate with global cooling trends. This impacted animal lineage and where they lived. In Africa, for example, some primates adapted to the expanding grasslands and savannas. And guess what? Among those primates were the ancestors of humans.
Wow, so our own lineage was shaped by these ancient climate shifts.
It's all connected, isn't it? From the Cambrian explosion to the rise of mammals, animal evolution has been this incredible journey of adaptation and diversification.
It's humbling to think about all that history. Now let's move on to animal body plans. This is where we really start exploring the blueprints for all the incredible animal forms we see.
Right, it's like looking at the architectural designs that make up the diversity of the animal kingdom. A body plan is basically a set of features, both structural and developmental, that define how an animal is put together. And I should clarify that the word plan doesn't mean there's some conscious designer. It's just a way to compare and understand different animal groups.
Like a framework for understanding their organization.
Exactly. And one of the most basic aspects of a body plan is symmetry, or in some cases, the lack of it. Some animals, like many sponges, are asymmetrical, their bodies are irregular, no real pattern.
Like freeform sculptures.
Kind of, yeah. Then we've got radial symmetry, where body parts are arranged around a central axis, like spokes on a wheel. Jellyfish, for example. If you slice them through the center, any way you cut it, you'd get mirror images. This type of symmetry works well for animals that are sessile, meaning attached to something, or for those that just drift around in the water.
Makes sense. They need to interact with their environment equally from all sides.
Exactly. And then we have bilateral symmetry, which is what we humans have, along with lots of other animals. It's the head-to-tail body plan. You can only cut it one way to get mirror images, a right and a left side. This type of symmetry is all about active movement.
Moving with a purpose.
Right. Most bilaterally symmetrical animals have what we call cephalization, meaning their sensory organs and brain are concentrated at the head end, and they usually have a central nervous system running down the length of their body. This lets them coordinate their movements and be more efficient hunters and foragers. Think of worms, insects, mammals, all bilaterally symmetrical. And these types of symmetry, they're ancient, going back at least 550 million years.
Wow, so these basic body plans have been around for a very long time. Now let's talk about the inside of these animals, their internal organization. The chapter mentions tissues again, but this time it's about how they develop from the embryo.
Right, so sponges, they went their own way early on and don't have true tissues. But all other animals, the metazoans, they've got them. And during gastrulation, that process we talked about where the embryo folds in on itself, it develops distinct layers of tissue called germ layers. The ectoderm is the outermost layer, and it goes on to form things like skin and the nervous system. The endoderm, the innermost layer, lines the digestive tract. And some animals, like jellyfish and corals, are diploblastic, meaning they only develop these two germ layers.
So 2 layers for some, and what about the rest?
The vast majority of animals, including all the bilaterally symmetrical ones, are triploblastic. They develop a third germ layer called the mesoderm, which forms between the ectoderm and endoderm. And this mesoderm, it's a game changer. It gives rise to muscles, connective tissues, and lots of internal organs. It allows for a whole new level of complexity.
Okay, 3 layers is the magic number for more complex body plans. Now, the next part can be a little confusing. Body cavities. We've got the coelom, the hemicolor, acolymates. It sounds a bit like alphabet soup.
It can seem that way, yeah. But basically, body cavities are fluid-filled or air-filled spaces within the body. They can provide support, help transport things around, and give internal organs space to move. A true coelom is a body cavity completely lined by tissue derived from the mesoderm. Animals with this are called coelomates. The mesoderm lining helps cushion and support those internal organs.
So it's like having internal scaffolding.
Exactly. Now, a hemicole is also a body cavity, but it's not fully lined by mesoderm. It's more like a space between the mesoderm and endoderm, filled with a fluid called hemolymph. This is typical of animals with open circulatory systems, like insects and many mollusks, where the blood or hemolymph isn't always contained in vessels.
So it's like a more open system of circulation.
Right. Sometimes animals with a hemicole are called the pseudocoelomates, meaning false coelom. But the thing is, coeloms and hemicoles have evolved independently, so using pseudocoelom to describe evolutionary relationships can be a bit misleading.
It's more complex than it seems at first glance.
It is. Many animals, like mollusks, even have both a hemicole for circulation and a reduced coelomer around specific organs, like the heart.
Okay, so it's not always one or the other.
Nope. And then we've got the acolymates, which are animals with no body cavity at all. These are usually flat, like flatworms. They don't need a fancy internal transport system because they can exchange nutrients and waste directly through their skin.
So body cavities are all about how animals manage their insides and get things where they need to go. Now let's talk about a really fundamental split in the animal kingdom. Protostomes versus deuterostomes.
That's a big one. It has to do with how they develop. You can broadly classify lots of animal phyla based on whether they're protostomes or deuterostomes. And the main differences are in their early development, specifically cleavage, how they form their column, and what happens to the blastopore.
Break it down for us.
Okay, so in protostome development, which you see in animals like mollusks, annelids, and arthropods, the early cell divisions, or cleavage, are often spiral and determinate. Spiral meaning the cells divide at angles, making a spiral pattern. Determinate meaning the fate of each cell is decided early on. If you remove one of those early cells, it can't develop into a whole embryo.
So those cells have a predetermined destiny.
Yeah. And in protostomes, the column, if they have one, forms from splits within solid masses of mesoderm. But the most important difference is what happens to the blastopore. In protostomes, it usually becomes the mouth. Hence the name protostome, meaning first mouth.
Makes sense. Now, deuterostomes, like us, are different.
They are. Deuterostomes, which include echinoderms and chordates, have radial and indeterminate cleavage. Radial cleavage means the cells divide in a more organized way, like slices in a pie. And indeterminate means those Ernie cells can still become anything. If you separate them, they can each develop into a complete embryo.
Like identical twins.
Exactly. And in deuterostomes, the coelom forms from outpocketings of the archenteron, that primitive gut. And here's a key difference. The blastopore becomes the anus, and the mouth forms later. Deuterostome means second mouth.
Fascinating. So it's all about which end develops first. It seems amazing that these subtle differences early on can have such huge consequences for the whole animal kingdom.
Right. It's a great example of how evolution works on these fundamental developmental processes. Okay, let's wrap up with the chapter's final section, which is about animal phylogeny. This is all about how we understand the relationships between different animal groups, and it seems like it's constantly changing as we learn more.
It's like a giant family tree, always being updated.
That's a great way to think about it. Animal phylogeny is a really active area of research. Scientists use all sorts of data, genomes, morphology, molecular data from different genes, all to piece together this evolutionary history.
And what are some of the key takeaways from our current understanding?
Well, one big one is that all animals share a single common ancestor, making the group Metazoa monophyletic. Basically, we all come from the same ancient ancestor. Another important finding is that sponges, those simple filter feeders, are considered the sister group to all other animals. This means they were the earliest branch to diverge from the main animal lineage.
So sponges are at the very base of the tree.
They are, and we have multiple lines of evidence for this. Fossil steroids from sponges in ancient rocks, molecular clock analyses, the fact that their collar cells resemble the closest living relatives of animals, and the fact that they lack true tissues. It all points to sponges as the earliest group.
Okay, sponges first. What's next?
The next major split leads to the Eumetazoa, which includes all animals with true tissues. This group encompasses pretty much everyone except sponges and a few other early branches like comb jellies and chirodarians. Then we've got the Bilateria, which are bilaterally symmetrical and have 3 germ layers. The Cambrian explosion, that burst of diversification, mainly happened within this bilaterian group.
Within the bilaterians, we have even more divisions, leading to the incredible diversity we see today.
Right. Exactly. Bilateria split into 3 main lineages: Deuterostomia, Lophotrochozoa, and Ecdysozoa. With the exception of chordates, which include us vertebrates, the other 2 groups are all invertebrates. Deuterostomia, it's named for the deuterostome pattern of development, where the anus forms first. They include hemichordates, like acorn worms, aerotonoderms, like starfish, and of course chordates. Then there's Lophotrochozoa, and their name comes from 2 features: the lophophore, which is a feeding structure, and the trochophore larva, a developmental stage. This group includes animals like ectoprocs, mollusks, and annelids. Finally, we have ecdysozoa, which includes things like nematodes and arthropods, and they're united by the process of ecdysis, or molting. They shed their exoskeletons or cuticles.
Wow, that's a lot of branches on the tree of life. And it sounds like scientists are still figuring out all the details. Are there any big debates going on in animal phylogeny?
Oh yeah, definitely. While we have a good overall picture, there are still some puzzles to solve. The chapter mentions 2 big ones. One is about comb jellies, or mechanophores. The current thinking is that sponges are the most basal animal group, but some studies suggest that comb jellies might actually be even more basal. They have tissues and a more complex body organization, which makes things tricky. The other debate is about a group called Acola, which are very simple flatworms. Some evidence suggests they might be the most basal bilaterians, meaning the earliest bilaterally symmetrical animal was pretty simple, but other studies disagree. These debates highlight how science is always refining our understanding.
So even the fundamental branching pattern of the animal kingdom is still being investigated. It shows how dynamic this field is.
Absolutely. It's not about being right or wrong. It's about using the best available evidence to build the most accurate picture we can. And as new data comes in, our understanding evolves.
Well, this has been an incredible deep dive into the animal kingdom. We've gone from the basic characteristics of animals, like their nutrition and tissues, to the major events in their evolutionary history, like the Cambrian explosion and the rise of mammals. We've explored their body plans, their symmetry, and their internal organization. And we've even touched on the ongoing debates about how we're all related.
We've covered a lot. Hopefully you, the listener, have a better understanding of how amazing and diverse animals are. Learning about their evolution and how they're all connected helps us appreciate our place within this incredible web of life.
It really puts things in perspective. Thinking about those small differences early in development that led to this vast array of forms and lifestyles is just mind-blowing.
It really makes you think, doesn't it? Given how certain features have evolved independently in different lineages, what other seemingly similar traits might have totally separate evolutionary histories? It's a question that keeps you curious about the intricacies of evolution.
Lots to ponder there. If anything sparked your interest today, we encourage you to dig deeper, check out the source material, and see what other mysteries you can uncover. There's a whole world of amazing discoveries out there waiting to be explored. Thanks for joining us on this deep dive.